So this third presentation is probably the one that is the basic in terms of method validation, verification, and transfer of analytical procedures. And I understand the audience varies from high school to college graduates, or even staff, or from industry, regulatory. So this is more of the most basic of all the three presentations. Again, before I make any presentation or presenting this topic, the disclaimer is that this presentation are my views and not the views or policies of the USP, FDA, or ICH. In terms of outline of my presentation, third one is simple because it will first describe the validation of analytical procedures, then verification of analytical procedures, and finally the transfer of analytical procedures. These are the things that I typically teach in pharmacy schools and graduate schools. So the general chapter validation, verification, and transfer. Again, this is a review. Validation is required when an analytical procedure is used to test a non-official article, or an official article is tested using an alternative procedure. So this is covering the general notices 6.3. General chapter 1226, which is the verification of compendial procedure. When is this required? This is required when the first time an official article is tested using a USP procedure. And finally, the ones that is general chapter 1224 would be the transfer of analytical procedure. And when do you use this? You'd use this when you are moving a non-compendial procedure, alternative or internal, moving it from one laboratory to another. So let's start with the validation of analytical procedure. So in analytical method validation is actually performing within the acceptable standards of reliability, accuracy, and precision for its intended purpose. So validation is the act of confirming that the method does what it intends to do. So also the validation guidelines in general chapters 1225 is a guideline, but as indicated here, validation activities is open to interpretation and practices differ greatly between organizations. Meaning, if there is a validation procedure from one organization, say one pharmaceutical lab A, they may be performing precision like for 12 samples, and for another laboratory, laboratory B, they may be performing precision using 6 samples or 12, 8 samples. So the details in terms of actual validation protocol varies from one organization unit to another. But the goals of validating analytical procedure are several-fold. Analytical procedures are of equal importance, meaning whether you are developing and validating a method for identification, or for assay, or for organic impurities, all of these have equal importance because they are intended to perform a specific function. And then in general, these validated analytical procedures are used in different ways for in-process testing, release, acceptance, even for stability testing. But again, you would want to have a validated method that minimize variation in terms of results. Again, the recurring theme for validation is it is suitable for its intended use. So the method is a planned systematic collection by the organization or by the applicant in terms of supporting their procedure. So this is again mentioned in the general chapter 1225 for those who are using the USP book, which is the monograph. There is no need to do validation simply because the procedures that are described in the USP book have been validated, and you can use them in another laboratory without doing any method transfer or transfer of analytical procedure. Definition of validation based on the United States FDA is consistent with USP and ICH. This is based on their document guidance for industry, which is focused on method validation, focus on the suitability for their intended use. ICH definition, again, is consistent. It is suitable for its intended purpose. But I wanted to clarify a terminology called the specification. So specification is referring to the analytical procedure and acceptance criteria. Many people refer specification as the acceptance criteria only. It has to include the analytical procedure and acceptance criteria together. So specification also is important to understand, because when we go to trying to understand verification, specifications are chosen to confirm the quality of a drug substance and drug product. It is not intended to describe the full characterization of the article, and it is focused only so that it ensures the safety and efficacy of the drug substance and the drug product. So that's the purpose for specification. Now, these are the lists of what is termed analytical performance characteristics: specificity, accuracy, precision. Some of you are familiar with these things, but let me just go quickly through it so that we have a common understanding, and we know what we're talking about. Specificity would be discriminating between the analytes of interest and the other components. What other components? So if you have a sample, for example, a drug product, there may be, when you put them in solution, there may be some impurities, degradation products, there may be some excipients in there that could interfere in your analysis. So specificity is important so that what you are trying to measure and what you actually see as a signal from your instrument is actually what you're trying to monitor. So the response observed is due to the single analyte of interest. Again, we are in the age where we have a more sophisticated instrumentation, and even at times, even optical isomers need to be isolated and separated because they have different um, uh, biological activities. The degree of specificity testing varies depending on the method type and the stage of the validation, and that specificity should be evaluated continually through the drug development process. So I showed you the drug development process earlier in a graph, all the way from the development up to the product approval, even to post manufacturing. So specificity needs to be continually evaluated, because at times there are impurities, degradation impurities that shows up, and they may interfere in terms of the specificity of your method. The accuracy is simple. This is an agreement between the accepted true value or a reference value against the actual results obtained. So either you know exactly what it contains, or you are using a reference material. And then accuracy, how do you do it in the validation? You actually do the accuracy by spike and recovery of samples. In terms of the definition of precision between ICH and USP, ICH have three things about precision. One would be the repeatability. And it's the same definition for USP, which is the same laboratory for a short period of time, same analyst, and same equipment. ICH also uses the term intermediate precision, which is within the laboratory, different days, different analysts with equipment on the same laboratory. And then the ones that are not typically done would be the reproducibility, which is what is termed interlaboratory. This would be, for example, analysis from a one laboratory, say for example at the FDA, or another analysis done in a academic, and then the third one would be something from an industry. So those are three different labs. Reproducibility. Make sure that we understand the term robustness and ruggedness, because robustness, again, is a measure of performance of a method when you are changing or deliberately changing making a small change in terms of the method condition. These method conditions that are being changed are the ones that are identified to be critical. Ruggedness, however, is a precision term. It is similar to reproducibility. These are different labs and different instruments, and they are not being widely used because it results in confusion at times. So definition of linearity between ICH and USP is the same. These are the results with which directly proportional to the concentration. However, USP has a little bit of a terminology because these are results that are directly or by a well-defined mathematical transformation. So at times, you may need to change some of the or transform some of the signals in, for example, or the concentration in terms of logarithmic. There are semi-log linearity curves. So it's not necessarily only signal versus concentrations. At times you need to do a semi-log. So that is the definition of linearity. Semi-log versus signal is applied, for example, if you're using an ion selective electrodes. Definition of linear range. If you have completed the linearity, you would want to uh, know the interval which between the upper and lower concentration is, and this is termed as range. In terms of limit of detection, same consistent definition between ICH and USP, which is this would be the lowest amount of analyte in a sample which can be detected. So this is not necessarily within the linear range. So, but it would be a signal that you can detect. At times, the limit of detection is close to the signal to noise ratio, somewhere between 2, 3, or even higher. Limit of quantitation, however, is different because limit of quantitation, as most of you know, would be the lowest amount of analyte in a sample that can be quantitated. So limit of quantitation would be, if you have a linear range, that would be the lowest concentration or the low range. So the limit of quantitation means that you are still in the range where the signal is proportional to the concentration. Data elements required for the validation is described in this slide. Now, in USP, it can be categorized into 4 different categories. The first category would be focused on the major components, and these are termed the assay. Category 2 would be impurity tests. So impurities can either be organic, inorganic, residual solvents, elemental analysis. So category 2 are focused on impurities in drug substance and drug products. Category 3 for data elements required would be what is termed the performance test. This is applicable to drug products. These are tests such as the dissolution test, disintegration, drug release, particle size. And then the 4th category is the term identification test. So it identifies the active ingredients. This slide describes to you the data elements required for the validation if you're doing a validation based on USP General Chapter 1225. Again, it depends on the analytical performance characteristics, and also it is dependent on the category type. As you can see, for example, if you are validating a category 1 assay, everything here listed is needed except the detection limit and quantitation limit. And so you would apply the same way to read this table. So as in quantitative impurity test, validation is required for everything except detection limit, but you need quantitation limit. It is not applicable for category 3 performance because performance is only applied to drug products, not drug substance. But here, the precision is applicable for drug products. Category 5, which is identification, everything is not needed except specificity. So characteristics are the same that are described by the ICH and USP. All these 7 validation characteristics are consistent, except in ICH, their category for identification and the required analytical performance characteristics is the same as that of USP. Impurities, again, the same for both the quantitative and limit tests, except for the assay. Their assay includes also performance tests such as the dissolution, content uniformity, and potency. So they bunch together the assay and the performance tests versus USP separates them in the previous table that I have shown you. So that would be a difference between ICH and USP. Critical step, first step in the validation. It is important that you need to know the purpose of the method. So you need to clearly state that. And per ICHQ2, the objective of the analytical procedure must be clearly understood. So there must be a statement of the validation conditions. Uh, so typically, when you do validation, it is assumed that you have already performed method development. In years ago, method development is something that doesn't need to be documented. It has become important that method development is a requirement and you need to document all your method development before you do all the validation. It is good to document all those method development because when you have difficulty with your methodology in analyzing your sample and you have to go back and analyze the conditions, what you have to change. For example, you're trying to do an alternative procedure. It is good to have a background information in how you develop the method. If the conditions are found to be mitigating against using the method, they should be indicated, meaning specific instructions such as analyze the sample within 2 hours, or you cannot go beyond plus or minus zero point 0.2 in terms of pH. Those are the specific items that needs to be included in the validation. The stability indicating method is something that the United States FDA has required for many of the pharmaceutical industry. Stability indicating methods are, as a term, it's able to monitor the stability of the drug product and drug substances. Now, for many years, the analytical method of choice is, for example, TLC or titration, and these are not stability indicating procedure. For example, if it's an acid-base titration, the base will titrate any acid regardless of what type of acid it is, as long as it has the acid functionality. While in HPLC, that's the reason why it's being used so much widely for many years, the HPLC allows you to do separation of the components before the quantitation. To demonstrate the specificity of a stability indicating procedure, all known interferences must be tested, or you can even develop a protocol to have a laboratory stress condition, meaning you stress the sample by a different condition, adding peroxide, adding acid base, adding light, and so on and so forth. So you can accelerate the temperature and humidity condition of your sample. This is to force your sample to degrade so that you can monitor the degradants while still maintaining specificity. So the validation data should indicate robustness evaluation. As I have mentioned, robustness is something that is done mainly at the method development stage. Nowadays, it has been mentioned in the USP General Chapter 1225, which is the validation. Also, when you're submitting your dossier to the FDA or USP, robustness data is now required. So we go to the verification of analytical procedure. Verification is actually not the same as validation because in the verification, validation is challenging the analytical method using a well-defined sample, while verification challenges the analytical environment using a well-defined method. So the applicability would be for drug substance, drug products, and excipients, The application can be on different analytical methodologies, titration, chromatographic, spectroscopic, and the definition of the verification, it is the confirmation by examination and provision of an objective evidence that the specified requirements has been fulfilled. Okay, and requirements, this is based on the Code of Federal Regulation. It is not actually the same as validation because only selected analytical performance characteristics are chosen so as to generate the appropriate relevant data. It is not a repetition of validation. So verification is needed so as to make sure that the laboratory performing the test can reliably perform with precision and accuracy the method as it is intended. So verification, uh, there are no general protocol for verification. This is one thing that whenever I teach this in other Asian countries, there's a lot of misunderstanding in terms of verification. But the guidance in the general chapter 1, 2 to 6 verification is only a guidance and it's not specific. I will, however, provide you with a recommendation on how to do verification, and this would be more or less targeted by how the US FDA expects the pharmaceutical company to do verification. Based on the GMP, it says that the suitability of all testing methods used shall be verified under actual condition of use. Also, verification is done only at the beginning, the first time. It is not done on a routine basis because once a method is verified that it is capable of analyzing the samples, then that would be it. Verification requirements should be based on assessment in terms of the complexity of the procedure and material. Now, when we were discussing about the validation, which is I mentioned about internal factors, which is focused on the analytical performance characteristics. The general chapter 1, 2 to 0, which is a holistic approach in terms of the validation, verification, and transfer, looks at everything. What is everything? And the environment that is referred to in the verification is focused on procedure related. What are those procedure related? Those are dependent on the analyst level of training, laboratory that is being used, the type of procedure associated to the equipment. Second thing would be it is material related. Sample, article to be tested, the reagents, the matrix. So these two uh, external factors are considered in the verification process. If this is the table that describes to you the data elements for validation, this is the one that I was going to provide you as a recommendation for to perform verification. But this first slide would be for drug substances and excipients. So it would depend on the analytical method that is being used. It is also dependent on the category of tests, whether it's category 1 assay, 2, 3, and 4. So for example, if you are doing a verification of the product using the assay method and your analytical method is HPLC, you only need to do precision. That's all you need to do. For category 2, for impurities, for a quantitative method using using an HPLC method, you have to do precision, specificity, and limit of quantitation. Same thing with the category 4 identification, but category 3 performance for drug substance and drug excipients is not applicable. However, this next slide, which is for drug products, now you have a category 3 performance verification. If you are using an HPLC, then you need to do precision. It would read the same way. So it would depend on, again, analytical method and the category of test that you are trying to verify. In terms of how does the industry define in terms of what do they do when we say precision, when we say accuracy? When they say accuracy in a verification mode, it is they determine the recovery at the specification range, meaning they need to know what is the specification range. I mentioned to you what is the definition of specification range, right? Earlier. Precision would be determine the repeatability only in the specification range. Specificity is a test for peak purity and matrix effect. Limit of detection is verification of detection at 50% of the specification. And finally, the limit of quantitation would be the verification of quantitation at 50% of the specification. Example of a verification of drug substance and drug products. And I will go through this one because this is something easier to see rather than the summarized two tables that I've shown you. For example, if you are doing a verification of a drug substance, such as the sodium chloride, your method is titrimetry. So precision may be needed. So that would be something that you only need to do. However, if you are doing a verification for a drug substance using an assay and your method is HPLC, then you need both precision and specificity. Now, if we go to the other categories for drug substances such as impurities, then you need to do precision, specificity, and limit of quantitation. Category 4, identification test, would be for a method that is IR, UV, or HPLC, then specificity is needed. If we go into the drug products for a category 1 assay using an HPLC method, then you need those three things: accuracy, precision, specificity. Category 2 for impurities using an HPLC method would require precision, specificity, and maybe limit of quantitation. For performance methods such as dissolution, you need to do precision. And finally, for category 4 identification tests, again, focus on the specificity. So to summarize verification, the intent of this chapter is to provide general information to laboratories on the verification of compendial procedure that is coming from USP that's being performed for the first time so that as to yield the acceptable results. It is not intended for retroactive, meaning if you have done it for the first time, you don't have to keep on doing it. And also verification consists of only selecting the analytical performance characteristics as opposed to doing everything in general in the validation. Finally, the last part would be the transfer of analytical procedure or method transfer. This would be general chapter 1, 2 to 4, transfer of analytical procedure, which is the last general chapter that was added in USP. And this is, again, the transferring unit is moving the method to a receiving unit so as to ensure that the receiving unit has the procedural knowledge and ability to perform the transferred analytical procedure as intended. And this is not applicable to USP monograph because you can use USP monograph at any time in any other lab without doing any formal transfer. So when is transfer needed? When, for example, you are moving a procedure or method from R&D to QC lab, site A to site B, QC to contract lab, instrument A, you're changing to instrument B. And then again, it is a risk-based approach because it depends on external factors, experience and knowledge of the analyst, specification of the product, the complexity of the analytical procedure. So you need to evaluate the extent and the type of analytical transfer procedure that you're going to implement. I mentioned there are 4 of them. So it all depends on whether they are validated or not. You've seen this slide earlier that a transfer waiver can be done, a revalidation, or a comparative testing. If it is not validated, you can do a co-validation with the transferring unit. Just to summarize, transfer waiver, what are those? Those are for new product composition that is comparable to that of an existing product. So there's really no change in the product. Also, the procedure transfer is the same, meaning that your staff moving from one site to another and the same equipment is being used. So a transfer waiver is possible to do that. Comparative testing, however, which is the most common transfer analytical procedure protocol, require the analysis of a predetermined number of samples from the same lot by both the transferring and receiving unit. Revalidation would be something repeating some or all of the validation experiments, but this would be the option that has the most time consuming, because it's, again, the receiving laboratory is going to be performing the validation. The co-validation is a bit better because it is both a collaboration between the transferring unit doing some of the validation, and then the receiving, which will also do some of the validation. This is used when procedure is not fully validated. So I have listed some of the resources that some of you would want to pursue and find out more. And these are available online. And the last slide actually lists down all this relevant information. The FDA, coming from the United States Food and Drug Administration, ICH, which is the international regulatory, provides international regulatory guidelines for drug substance and drug products. And also, if you have the book for USP, you can refer to some of this general chapter. Thank you very much for your attention. It's my honor to present all these three topics, and I hope you learned something from it.